Hey everybody, welcome back. This is week 31. This is our last week of new material, so you'll only have textbook questions on one lesson, and then the additional homework this week is going to be reviewing body systems because that'll also be on the last test. So let's get started. The last lesson is genetics, and it's why you look like what you do. Basically, we're going to explain, we've always talked about the nucleus and the fact that all the information is stored there. We're going to kind of dive into that a little bit more deeply in this section. You'll cover it pretty extensively in biology, where you'll learn about Punnett squares and how to actually do Mendelian genetics. And it'll cover it even more if you end up taking anatomy and physiology in high school. Okay, so genes are tiny bits of information that are contained in the DNA, and basically there are millions and millions of groups of nucleotides that you've already in your DNA, and each group of those is a gene that encodes for something special. You get half of your DNA from your father and half from your mother, and those form together in a zygote. That zygote has all of the DNA needed to make all the cells in your body. These genes determine skin color, hair and eye color, what your hair is going to be like, if it's curly or straight, what your facial features are. Something kind of interesting that you don't think about, 99.5% or more of all of our genes are the same as other humans. We even have like 97% of our DNA in common with chickens. So quite a bit of the genes have to do with protein formation, with making the different kinds of blood components, bone cell structure, all of that, which is very common across all mammals. Okay, dominant versus recessive genes, you've probably heard these terms before, and that's the idea that you get two full sets of DNA in every cell of your body. One set came from your mom, your birth mom, the other set came from your birth dad, and these sets have matching places where they're found in the chromosomes. And because of that, for each gene, there's a dominant and a recessive one. And if you have one of each, the dominant one will prevail. So for brown eyes and blue eyes, that's kind of a weaker example. It's actually more complicated than a single gene. But just to keep it something that you're familiar with, the idea is if you have even one of your parents gave you the brown-eyed gene, you'll be brown-eyed. They both have to give you their blue-eyed gene to be blue-eyed. So if, for example, if looking at the box on the left, if you have a brown-eyed gene from one parent and a blue-eyed gene from the other parent, your eyes will be brown. Okay, and that just kind of explains that. Okay, there are different people groups, but not races, if you think about it in terms of genetics. So genetically, all people have the full amount of, I guess, variety in them. So God created Adam and Eve. All humans came from Adam and Eve, and there are genes that have a lot of variety in them. Okay, some of the information was lost during the worldwide flood, but all of Noah's genes, family's genes, got preserved. And interestingly enough, it is likely that God knew way more about genetics since he created it than we knew even up until the last few years. And so it's likely that each of Noah's wives had a very, very different gene pool from each other, just to allow there to be the maximum amount of genetic diversity after the flood compared to what it could have been if it had all just been a single family. So one family was for sure preserved, and that was Noah's, but also 3 other bloodlines got preserved as well, because each of Noah's sons' wives had different folk in that got carried on to their children. After Tower of Babel is when people spread throughout the earth, and there were often similar characteristics to each other. This could have been environmentally controlled, but it also could have been just the diversity in the gene pool from Noah's sons' wives. Okay, DNA is the genetic code that is inside every cell of your body. DNA stands for deoxyribonucleic acid, and it's a very long molecule that codes for every part of your body. Interesting thing about this word, you guys are going to hate me, but even though spelling hardly ever counts for tests, you actually, to get full credit for this one, you do have to be able to spell it. Partly because I think it's actually a fairly easy word to spell if you can pronounce it. The vowels are pretty straightforward, and by learning to spell it, you're sure going to incorporate every single one of the syllables. So D, oxy, you can remember that the Y begins with, the E sound in oxy is a Y because it's actually short for oxygen. It has to do with oxygen being one of the molecules in DNA. So deoxyribonucleic and then the word acid. Okay, so you may want to start spelling that. Oddly enough, it's one of the very, very few questions that's never missed on the test. Almost everybody gets the spelling right. It was discovered about 70 years ago, and they were actually able to describe it as a twisted ladder called a double helix. And the word helix is basically, it means sort of something around curvature. One way you can think about it is if you think about a slinky, the wire in a slinky that goes round and round and curves up. It's a single string, but it's all wrapped up. That's a helix. Another example of a helix is if you follow the thread of a screw around, that it's traveling up, but it's still a single line. Okay? So here's the structure of DNA. If it's unwrapped, it looks like a ladder. And so the phosphate molecules are like the sides of the ladder, and the rungs of the ladder are the sugar molecules called deoxyribose. And what they are is they are a series, and depending on what these letters are, I know you can see the A, the T, the C, and the G. Depending on what those are, that's what determines what the traits are. So they're called base pairs because they always match up 2 to 2. There are 4 different types of bases: adenine, thymine, guanine, and cytosine. Okay. The good news is this is the only word bank you're going to get on the last test. So you will be given those 4 words: adenine, thymine, guanine, and cytosine. And they're normally, when they're described in DNA molecules, they're just abbreviated with their capital letter that they start with. And there are 2 bases connected together for each rung. A can only connect with T. They only mate with each other, and that's why when you look at it, it could be on either side, but they can only connect with each other. G only connects with C. You do have to know which ones mate together, and the way I always remember it, if you think about it alphabetically, is the one at the beginning and the one at the end are together, and the 2 letters in the middle are together. So adenine and thymine are together, and guanine and cytosine. Okay, and their sequence of base pairs that are wrapped up, sometimes they're described by their capital letters, other times they can be described by colors. So genes can be anywhere from 3 or 4 base pairs strung together to a couple of hundred strung together. Okay, but the pattern of them is what makes you unique. It also is what makes you all the same too. So the way that your body synthesizes protein, the way your pancreas releases insulin, the way your neurons fire, the way your blood clots, all of those things are, almost everybody has exactly the same. Okay, each gene contains about I was wrong about the number, 100,000 base pairs. Some can contain up into the millions. Okay, the Human Genome Project, you may have heard of that over the years. Basically, in the early 90s, a group of scientists all over the world decided to map out the base pairs, but they had to divvy them up among the chromosomes, and each chromosome is pretty long as well. So what they did is they gave each person a different assignment to study what the pairs likely were. And it took over 13 years, and then it took a few years after that for somebody to assemble them all. Okay, and they found that there's about 3.3 billion base pairs and about 21,000 genes. Okay. Chromosomes and cell division. This is just a very, very quick overview on how it works. Uh, Basically, the chromosome that you see there, it looks almost like a little four-legged spider or something. That's only what it looks like during cell division. Most of the time, all of your DNA just looks like a giant bowl of spaghetti noodles floating around in your in each of the nucleuses of your cells. It's only right before it's ready to divide up, it'll form into the chromosome shapes that you see at the bottom of the screen there, and that's in order to be ready to separate. So this kind of reproduction is called meiosis. I'm sorry, mitosis. I always get those two mixed up. And it happens in all the cells in your body. So basically, all the chromosomes line up and they're duplicated, and then they split, and each piece goes into a separate one. And then so after they get split, then they can separate, and so that one can go into each of the two new cells. When it's floating around in your cells, the main job of DNA is to make the proteins, and the proteins are the building blocks of all the cells in your body. So when we were studying proteins and starches and fats and all of that, the starches and the fats were purely an energy source for the cells to be able to do metabolism. But all of the actual cells are made with proteins. And so basically, to make a protein, whatever piece of the DNA strand, that big giant pile of spaghetti noodles in your nucleus, is targeted. And then there's a protein in the cell that unzips the section. And then it makes a copy of that section. And then it zips it back to normal. And it uses that copy to create the needed protein. So it never takes away from the DNA. It makes a copy of it and uses the copy. Okay, we'll talk a little bit about mutations. The idea of a mutation is when there's something that happens in one of those nucleobase tied pairs that can change things up. So the first thing that's important to understand, and there's a video about this on your assignment sheet this week, is that you have two complete sets of DNA in your body, one from your mom and one from your birth mom and one from your birth dad. If there's a flaw or a mutation in one section from one parent, usually almost always the mutations are recessive. So what that means is that the code from the other parent will dominate, so the flaw isn't passed on to the baby. And so if there's a missing section or just an incomplete section or an incorrect section, then your body almost always takes the one from the good parent. If there's a mutation in the same location of both codes, then the child can inherit the birth defect or the disease from the parents. And that's why, especially if there's a history of older siblings having something, some parents may get some genetic testing done just to kind of find out more specifically what's going on. Okay. The idea behind evolution is you go from atoms to molecules, and then molecules to very simple life forms, and then on to monkeys and on to man is the idea. And so their idea is that the very, very most basic bacteria turned into a protista, and that advanced farther and farther along until we got to the complex kinds of life forms we have today. In general, mutations are almost always harmful to the organism, and they don't typically add genetic information. And that's why it's hard to explain or even theorize the idea that you can add additional genetic information through mutations, which is the premise behind biological evolution. Gregor Mendel is one of the most famous scientists of all time. He was actually a monk, and he was also super into gardening and things, but he decided to try and trace characteristics from different generations. It's hard to do it in people because the generations, to even get a one generation jump, takes 25 or 30 years. With plants, the life cycle can be a year or less, so it's a lot easier to study. And if you breed them properly in greenhouses and stuff, you can actually get through 5 or 10 generations of plants in less than a year. So anyway, he lived in Moravia, and he loved nature. And he taught, as a lot of monks did, and priests and nuns back in the 1800s, he was also taught in private school. And his main studies were on pea plants. And kind of what he found is that if he crossed a tall plant with a short plant, he would, especially if it was a certain tall plant, he would get two tall plants. But if he crossed two tall plants together, he could occasionally get a short plant. And so he did the same thing with seed color, seed shape, and the type of seed, the color of the seed coat, and with the pod color and the pod shape. And that's how, and he did it over and over and over again. He was very careful to isolate. He also kept really, really good records. And he crossbred over 28,000 plants, and that's when he discovered that whole concept of dominant and recessive genes, and it earned him the title Father of Genetics. And these are the laws of inheritance that he published, and they're pretty well been confirmed with all of the studies since then. Hereditary factors do not combine, they are passed intact. What that means is if you inherit a tall gene from one parent and a short gene from another parent, it doesn't make a medium gene. It doesn't work like that. Your body will choose, or your DNA will choose one over the other to exhibit. And almost all genes have a dominant gene and a recessive gene, and you might be getting the same one from both parents or two different ones, one from each parent. If you get a different one from each parent, then the dominant one will be what your body will show. Independent of sort. This is my favorite one, and I am so thankful this. Basically, because your parents have two sets of DNA, you never know which one they're going to be giving to you. And it's actually even, it's not even all or nothing. It's different pieces of their DNA can go in. And that's why you're not exactly like your brothers and sisters, because you're getting different pieces of their DNA. The only time you get the exact same DNA from both parents is identical multiple births, like identical twins, identical triplets, etc. Okay. And the Punnett square you'll be learning about in biology, so we won't go over it right now. It just explains how the combination of which genes your parents pass on are reflected in not only what you exhibit in your body, but what you carry to pass on to your children. Okay. They use it a lot to do genetic testing for plants and animals. It's especially used a lot in like breeding cattle and things like that to try and pass on things that are good for the cattle industry and chickens and all that kind of stuff. Okay, so let's do some questions. What do you call the tiny bits of information? And each tiny bit of information is a gene. Two sets of genes come from, that's an easy one. One from your mom and one from your dad. And by this we mean birth moms and birth dads. The stronger gene is called the dominant gene. The weaker gene is called the recessive gene. And there's one race of people. All are offspring of Adam and Eve. All are also offspring of Noah and his wife. The different people groups are caused for a couple things. One is there was a lot even before the flood with Noah's sons' wives, but also after the Tower of Babel, there was a lot of dispersion into different parts of the world. Okay, before I put it up here, I want you to try and spell it. So I'm going to give you a sec to think about how to spell it. You can spell it out loud if you want. Deoxyribonucleic acid. The three molecules that make up the DNA molecule are a sugar, a phosphate, and a base. When they coil up and they're ready for cell division, they're called chromosomes. And if there's a mistake in the DNA code somewhere, it's called a mutation. The father of genetics is Gregory Mendel. And he studied pea plants. and traits passed down by parents. Okay. This is the last PowerPoint presentation we're going to be doing. For week 32, since there won't be a PowerPoint and there won't be any new material, there's going to be an optional live class to review for test 5, and we'll be using the new meeting software that they sent out to all the humanities teachers. And I will announce that when and where that, well, where that's going to be. I will also, like I said, I'll let you know in the week 32 handout when that's going to be. It'll be later on in the week before the test. Hope you guys enjoy this. There's some great PowerPoints explaining DNA, some really good animations this week. It should be a fairly easy week. I think I told you last week I thought everyone did great on the chapter 4 test. I mean on the test 4. The next test I'll be about the same length, but it'll be a little easier. There won't be all the complicated blood flow stuff. It'll mostly be vocabulary. So anyway, I hope everyone has a great week, and I will see you next week.